Alrighty guys, it's Miss Hendricks and this is Unit 1.6 over Hardy-Weinberg equilibrium. So we've talked about Hardy-Weinberg before, and we're just going to do a little bit of review before we get into it. So you should have read/watched the previous videos, and they should have given you information about different types of evidence of evolution. So in your reading, you read about the fossil record, right? And if you didn't know, we use something called carbon-14 to date our fossils to figure out how old they are based on the decay of carbon. It's radioactive and it decays in a certain number of years. So we use carbon-14 dating for that. You should have learned about homology and analogy and the difference between those two. So homology meaning they have a common ancestor, analogy meaning that the structures are just similar. And lastly, you should have learned about molecular evidence, so DNA. So that's something that Charles Darwin did not have back in his time, and it's something that further helps give evidence for evolution. If you're given molecular evidence, that is the end-all, be-all. That is the most important thing to base your evidence of evolution on. So that one is obviously the winner of all of those. So today we're going to talk about Hardy-Weinberg equilibrium. This was in your first reading for a little bit, and a really good definition for that would just be a population's allele and genotype frequencies remaining constant unless there's some type of evolutionary force acting upon it. So Hardy-Weinberg is kind of a theoretical, mathematical way to see if evolution is occurring or if it's not in a population. Now you might think, of course, evolution is occurring. Why would we even want to test this? And this is so that we have something to compare what we observe in nature to. So we will have our observed values that we see in nature. If those observed values match what we calculate with our Hardy-Weinberg equilibrium equation, then we would say that there is not evolution occurring. Maybe the ecosystem is very stable and there's not evolution occurring. Versus if the numbers we get in the Hardy-Weinberg equilibrium equations are different than those that we observe in nature, then we would say that evolution is occurring. There are different variables that we need to talk about that are important to determine Hardy-Weinberg. The first one would be our p-value. So if you can think back to genetics in IA, p is the allele frequencies of the dominant allele, right? There's dominant alleles, there's recessive alleles, and our recessive alleles are q values. That's the allele frequency of our recessive allele. So if you were given the frequency where you have 10 dominant alleles out of a total population of 50, how would you calculate the frequency? Well, you would just put the number of alleles that are dominant over the total number of alleles you have. So in this case, it would be 10 over 50, and 10 over 50 would give us which of these values? It would give us the value of 0.2, because 10 divided by 50 is 0.2. Remember, an allele is an alternate version of a gene. So if I was talking about the phenotype of attached earlobes and I was recessive for this specific trait, what would my genotype be? Remember, my genotype would have one allele from my mom and one allele from my dad. So if I was recessive for this trait, and we're going to call the letter E is going to be our letter for this, I would have little e, right? In order for it to be recessive, I have to have both alleles as that recessive gene. So how many alleles do I have total? I have 2 alleles. I have one little e from my mom and one little e from my dad. So each individual, you have to remember, has 2 alleles or 2 versions of that specific gene. So you need to remember what an allele is. I talked about it at the very beginning of this part of the section, so go back and review that if you missed it. There's also some really important terms we need to go over, and how that P and that Q applies to Hardy-Weinberg. So if I said that something was a homozygous dominant individual, that means that individual has 2 of the dominant alleles. So going back to the earlobes, they would be big E, big E. To apply this to Hardy-Weinberg, that would be my P squared value, my number of homozygous dominant individuals, not alleles, not the frequency of alleles like P was. This is the number of homozygous dominant individuals. Our heterozygous, they have 1 of each, right? They have 1 dominant and 1 recessive, so they would be big E, little e. In the case of Hardy-Weinberg, that would be our 2PQ value. And then lastly, for our homozygous recessive, this would be our individual with 2 recessive alleles like I had for the trait attached earlobe, so little e, and that would be our Q squared value. So there are specific steps I would follow when going through a Hardy-Weinberg problem. I've included the 2 Hardy-Weinberg equations that you would have on your formula sheet, and I'll show you where those are in class. So our 2 equations that we're going to use is P squared plus 2PQ plus Q squared equals 1, and P plus Q equals 1. And remember, P is the frequency, it says of allele 1 in a population, it's the dominant allele, and Q is the frequency of the allele 2 in a population, so the recessive allele. And I have steps here that I would go through, but before I explain why I go through the steps in this way, let's look at this example with our 2 bunny rabbits. So we have a brown rabbit and a white rabbit. If I was to tell you that the brown rabbit was our dominant trait and our white rabbit was our recessive trait, which bunny could you look at and you would specifically know for sure its genotype? Obviously, that would be the white bunny, right? If it's the recessive one, I know that its genotype is, let's say, R, right? It is recessive, so it has to have 2 of the lowercase letters or the recessive genes in order to express the color white. The brown bunny, on the other hand, could be big R, little r, or big R, right? It could be a heterozygous dominant, or it could be a homozygous dominant or a heterozygote, right? We don't know its specific genotype. So that's why for Hardy-Weinberg, you need to start with your Q squared value. Remember, Q squared is your homozygous recessive, right? So it'll either give you that outright, which would be great, or if they don't give you that value, you would need to calculate that by figuring out the number of homozygous recessive individuals out of your total number of individuals, right? So if I had 13 homozygous recessive individuals out of 100 in the population of bunnies, I would do 13 divided by 100 to figure out my Q squared value. Then to figure out my Q value, or the frequency of that allele, that would be taking the square root of that Q squared value. Remember, square root gets rid of a squared, and now I have my Q value, or the frequency of that specific allele. Now you can use that second equation, the P plus Q equals 1, to figure out your P value, right? You have your Q value, so you just need to do 1 minus Q to give you P. To get P squared, or your number of homozygous dominant individuals, you would just square your P value. And then lastly, to figure out your heterozygotes, you would just do 2 times your P value times your Q value, or 2PQ. If this doesn't make sense, we are going to go through some problems in a different video, and I can help you on that. And there's also a self-grading worksheet you can work through. So remember, Hardy-Weinberg assumes that the alleles and genotype frequencies are stable unless evolutionary forces are acting on that specific population. So there are those 5 assumptions that must be met in order for a population to be in equilibrium. That was no natural selection, no mutations occurring, no migration, so no immigration, moving in, or emigration, So moving out, it has to have a large population, and there needs to be random mating occurring. If any of these are getting violated, then evolution is occurring in that population. So one more thing before I wrap it up. When you get your values of the P and Q values, right, what do you do with them, right? You have these values, now what? Researchers, like I said at the very beginning of the video, will take these P and Q values and compare them to the ones that they actually observe in the population. So you calculated them using your Hardy-Weinberg equilibrium equations. You got that the genes should be split 0.5 and 0.5 for dominant and recessive, but then in the actual population, they noticed that it's 0.7 and 0.3, right? So the ones that you calculated did not match what they observed in nature, therefore the population is not in equilibrium. So remember, we're trying to determine if evolution is occurring or not. They look at the P and Q value over multiple years to determine if it's in equilibrium. Otherwise, they know the evolutionary forces are acting on that population. And make sure that you really understand each of the equations and what exactly each of those terms stand for, right? Don't get P squared and P confused. One of them, P squared, is for individuals and P is for allele frequencies. Don't confuse them when you're working through Hardy-Weinberg equations. Okay, if you have any other questions, please let me know. There's going to be a couple of Hardy-Weinberg equations to work through on this video, and if they're confusing, please just come talk to me or email me or whatever you need to do to get additional help on that. Don't forget to take chances, make mistakes, and get messy. Hope that was helpful.